Okay, hi everyone. This is our lecture on chapter 9, which is the cell cycle and cellular reproduction. This chapter also sort of starts a new unit within the textbook, right? Our last couple chapters were kind of focused on biochemistry, right? We're talking a lot about sort of metabolism, chemical reactions in general. We talked about enzymes, and then we particularly talked about photosynthesis and cellular respiration, which are all, biochemical pathways that are comprised of various different reactions. So we sort of switch gears now. And really, this whole unit is about the genetic basis of life, okay? So, like I said, we'll start with chapter 9. Make sure you have your next exam review sheet, which would be the exam 3 review sheet out, and you should be taking your notes on that review sheet as we go through. Okay. All right. So for our purposes, what we'll cover in this unit is chapters 9, 10, 11, and 12. We will not cover chapter 13 and chapter 14, just in case you kind of have your textbook out and are looking at that. All right, so let's get started. Okay, in terms of the outline, we're going to talk about the cell cycle in first, then we'll talk about chromosomes specifically. Then we will talk about the stages of mitosis and also cytokinesis. The stages of mitosis in terms of identifying them from pictures. We also go over that in lab. You guys do a lab activity that's tied to that. And on the lab quiz, there'll be a lot of identifying, like here's a picture of a cell, tell me what stage it's in. For the lecture exam, I won't put pictures like that. Okay, so I kind of try to separate things out. Then we'll talk about the cell cycle and its relationship to cancer. And then just literally, I think there's like 2 slides or maybe even 1 slide on prokaryotic cell division, really just to emphasize the point that in prokaryotes like bacteria, the way that they divide is not through mitosis, okay? All right, so what is the cell cycle, right? So the cell cycle says there is an orderly set of stages from the first division, right? So when the cell is first born, think of, to the time it's going to divide and produce daughter cells, okay? So that entire time is the sort of life of the cell, the cycle of the cell. So what's going to happen just prior to that next cell division is that the cell's going to grow larger. It's going to double its organelles, right? And it's going to replicate its DNA. Why? Because you're going from 1 cell to 2 cells. So you have to make sure you double your amount of DNA, you double the organelles. So each of those, that new cell has all of the components that it needs, okay? So if we're going to talk about the cell cycle and break it down into 2 major stages, okay? Interphase, and interphase itself includes several stages, and we'll talk about that, and the mitotic stage. So, okay, so the 2 major stages of the cell cycle are interphase and the mitotic stage. The mitotic stage, we don't just say mitosis because cytokinesis is not really part of mitosis, okay? So the mitotic stage is mitosis, right, and cytokinesis. So this is the cell division part of it. Interphase is sort of what's happening to prepare the cell to divide, okay? All right. So as I said, interphase is divided up into different stages as well. So we want to talk about those stages. Most of the time that the cell is going to spend in its sort of lifetime is in interphase, okay? That's when the cell is performing its usual functions. And as I said on the previous slide, it's going to prepare to divide, okay? Also important to note that the time that's spent in interphase is going to vary depending on cell type, okay? And another kind of thing to point out here, there are actually, many cells in the body that don't really, aren't really in the cell cycle, right? So they don't complete the cell cycle, meaning they're not going to divide, right? So nerve cells like neurons, muscle cells, they're not in the cell cycle. They remain in something we call the G0 stage, which is a quiescent or resting stage. And they're not actively dividing. So any cell type that's not actively dividing, right, producing new cells, is in this G0 stage, and sometimes we say they're not in the cell cycle, okay? So again, so that's that sort of exception here, okay? And then also, and cells that do divide, there will be differences in terms of how long interphase is, okay? All right, so just to kind of point that out, and again, there's a lot more details we can talk about within this, but this is, again, meant to be an overview. When we get into cell biology sophomore year, we do talk about the cell cycle in a lot more detail, okay? So if we're going to look at that interphase, right, and now we got to break that interphase up into the stages that make it up, right? So interphase is composed of G1, S, and G2 phases, okay? It says it takes up about 90% of the cell cycle. So if you're going to think about interphase and the mitotic stage, 90% of the time would be spent in interphase and then 10% in the mitotic stage, okay? So that's what that is saying here. What's happening in each one of these phases? So in G1, it says the cell's recovering from the previous division. It's going to double its organelles. It's going to grow in size. It will start getting ready for DNA synthesis because in S phase, DNA replication or DNA synthesis will occur, right? Meaning the cell is going to replicate its DNA. So it says proteins associated with DNA are synthesized. Talks a little bit about the form of the chromosomes, right? So chromosomes enter S phase with one chromatid each, but then they leave with two because, again, the DNA is replicated. Now, we'll talk a little bit more about sister chromatids and about chromosomes a little bit later on in the chapter, so don't worry about that right now. But, I mean, I don't know. Yeah, I guess you don't have to worry about understanding that specifically because I'm going to come back to it and explain it a little bit more. But bottom line is chromosomes have one chromatid, right? But once the DNA is replicated, you'll see two sister chromatids. That means that DNA is replicated. Okay. And then those sister chromatids will be attached, right, until they get to mitosis, and eventually they'll be separated during mitosis, okay? In the G2 phase, again, this is the order that they occur in too. We should know that, G1, S, and G2. In the G2 phase, basically, this phase is between DNA replication and mitosis, okay? The cell basically is getting ready to divide, right? It synthesizes proteins that are needed for mitosis, like microtubules, okay? And so that's one example, but many other proteins that are necessary for cell division as well. All right, so if we talk now about the mitotic stage, it's mitosis and cytokinesis, okay? So we will talk about the stages of mitosis, and as I said, in terms of identifying different cells in different stages, you'll do that in one of your lab exercises, okay? We'll look at, and usually we look at cells under the microscope ourselves in lab, but I'll show you pictures, right? Just like you were looking under the microscope to say, you know, basically kind of search around and be like, all right, I know this cell is in, you know, metaphase, this cell is in anaphase. So we'll talk about those stages in a little while, okay? So mitosis is nuclear division, okay? Daughter chromosomes, right? They line up in the middle of the cell and then they basically get separated by that mitotic spindle. And as I said, we will talk about the details of mitosis in a couple slides, okay? Cytokinesis is the division of the cytoplasm, okay? So again, cytokinesis is not part of mitosis. It's not a stage of mitosis. It always happens, you know, right after mitosis. It's sort of something that's related to mitosis, but it's not actually part of mitosis because mitosis is the division of the nucleus, okay? And so what happens after this mitotic stage? You have two genetically identical daughter cells, okay? All right, so if we look at this figure here, basically it's showing you the phases of the cell cycle, right? Remember, we talked about the two main stages being interphase, right? And the mitotic stage, okay? And then we can break down interphase into these three phases here, okay? So G1, S, and G2, we talked about that. Another thing to note, well, one here, this is showing you this G sub 0. Remember I mentioned that with basically cells that are not actively dividing, they'll be in this sort of resting stage. Now, if something would then came along to stimulate them to divide, they could then re-enter the cell cycle, okay? That's what this is showing you here. In terms of these little stop signs, right, these are checkpoints, okay? So it's important for the cell to check to make sure everything's okay before it moves on into that next phase, okay? So here it says cell cycle. This is the main checkpoint. If DNA is damaged, apoptosis will occur. Otherwise, the cell is committed to divide, okay? It says when growth signals are present. So as long, and nutrients are available. So as long as everything's favorable for the cell to divide, it will go on to divide. This checkpoint, so sort of checks for the availability of nutrients and things like that in the external environment. but also checks internally to make sure that there is no damage to the DNA, right? It would not make sense for the cell to replicate damaged DNA, okay? So if there's damage to the DNA, the cell would then enter apoptosis, which is cell death, and that's it, okay? It would undergo that programmed cell death. Again, if it went on to replicate its DNA, it's going to produce daughter cells that are defective, right? And that can ultimately lead to things like cancer and other problems. All right, so that's these checkpoints. So, okay, so then if we move along, remember S phase, right? We have DNA replication, S synthesis, think of that. If we now move into G2, it says growth. So all these G1 and G2, there's always growth going on. Here in G2, again, most of what's happening is growth, but it's preparing for the mitotic stage here. Okay, so it says here, it's another, another checkpoint here, mitosis checkpoint. It's not really, I don't really like the name mitosis checkpoint, to be honest. But the reason why the book is naming it that, because it's saying it's checking to make sure everything's ready for mitosis. Okay. So it says, again, it's checking to make sure now at this point that DNA was replicated properly in S phase, right? Again, if that DNA is not replicated properly, the cell doesn't want to go through that mitotic stage and produce two new daughter cells, okay? And so that's what this is saying here. Apoptosis will occur, again, if that DNA is damaged. Now, there's another checkpoint here, which is basically sort of in the middle of mitosis, and this is the spindle assembly checkpoint. And so basically, if the chromosomes aren't aligned properly, then the cell will undergo apoptosis. Same thing, because then the daughter cells will not get the correct chromosomes, which again will lead to dysfunction of the cell and can lead to things like cancer and other diseases as well. All right, so that's what this is showing here. So basically everything that's on this slide is what I talked about in the the previous slide or two. All right, so important to understand this, to understand the two main stages of the cell cycle, interphase and the mitotic stage. Also know the phases that, the three phases that make up interphase, right? G1, S, and G2, what's happening at those phases, and then understand what the purpose of these checkpoints is, right? Okay, now, again, this is something that we're not going to go over in great detail, but I have to mention it because it's super important. If you think about this cell cycle here, you might imagine that it's very important that this is under tight control, right? The cell has to control this, meaning that, if the cell cycle or cells are in the cell cycle when they shouldn't be and they're rapidly dividing, right? It leads to rapidly dividing cells or the production maybe of a tumor, right? If this isn't controlled enough in terms of it being maybe too slow or cells that are supposed to divide aren't, it could lead to problems with repair and growth and things like that, okay? So you can see how it has to be tightly regulated, right? So if it's sort of too much or too little, it leads to issues. Okay, so there are signals that control the cell cycle, okay? There are both external and internal signals. So I sort of mentioned the external signals on that last slide. And so those are things like growth factors. Okay, so these are things that are in the extracellular environment that might bind to a receptor on the plasma membrane and tell the cell, yes, there's lots of growth factors around, right? Those growth factors will then say you should divide, right? Things are favorable for you to divide. There are also internal signals, and these are there's one particular family of proteins called cyclins. Again, these are inside the cell, right? And these cyclins are there to sort of control that cell cycle, meaning moving from G1 to S into G2. That movement through those stages or phases is controlled by cyclins, okay? So it says specific cyclins must be present for the cell to proceed from one stage to the next, okay? If those cyclins aren't around, the cell will stop, right, at those checkpoints kind of and stop within some of these phases. So they are necessary to progress through that cell cycle. It says here, damage by solar radiation or X-rays repaired at the G2 checkpoint. I'm not sure why that's sort of, I guess that's kind of in a weird place there, but there is, if you look here at G2, right, if there was some kind of damage, one that was induced by replication or by radiation or something like that, potentially it could be repaired. So the cell might try to repair it. If it can't be repaired, then the cell would go through apoptosis. But if it can repair that damage, it would then progress through. Okay? All right. So I've been mentioning apoptosis, right? I also mentioned that it's called programmed cell death. Okay? There are a sort of specific sequence of events that occurs when the cell goes through apoptosis, and there are certain morphologic or features or sort of features that you can observe under the microscope that help sort of tell scientists that a cell is going on their apoptosis. Okay, and some of those things. So, like I said, you can see some of these things under the microscope, usually with the aid of some type of stain or something like that. But these are things that are observable. Okay, so, and some of the things are fragmenting of the nucleus, right? Blistering of the plasma membrane, and also, white blood cells or other cells would sort of can be around to kind of clear up those dead cell fragments. Okay. They are caused by a certain group of enzymes called caspases. So this is another way that scientists can identify that a cell is going through apoptosis, right? We could look for the presence of these specific enzymes, okay? It also says here mitosis and apoptosis are opposing forces. Think about it, right? Mitosis or cell division increases the number of cells. Apoptosis is cell death, is going to decrease the number of cells, okay? So you can kind of think of those two as being sort of opposites of one another. All right, so these are some of these sort of characteristic features that you see in apoptosis. So this is showing you, normal cells here. In terms of a cell that's going to start undergoing apoptosis, the cell is rounding, the nucleus starts to collapse here. Here it's showing you the chromatin is condensing, the nucleus is now broken up into pieces, it's fragmented. Then you start to see these blebs in the plasma membrane, right? And then basically finally you see that the cell is fragmented, right? You kind of see just DNA fragments, and this is actually showing you with an electron micrograph, you can see some of the cell fragments here. So like I said, you can observe this under the microscope. Okay, so hopefully we are okay with understanding that cell cycle, knowing the parts of the cell cycle, understanding that there are checkpoints, understanding that the cell needs to have tight control over that cell cycle, otherwise it will lead to issues, okay? Knowing those terms, apoptosis, what does that mean? Programmed cell death. In terms of the phases of mitosis, we didn't really talk about that yet, okay? So don't worry about that. But really we were just talking about that cell cycle in general, okay? A family of proteins that controls the cell cycle, right? An internal signal, we said, was called cyclins, okay? In terms of external signals, examples are growth factors. Those are things in the external or extracellular environment that indicate that conditions are favorable for that cell to divide. So again, if there's a limit or a limitation in the amount of resources outside the cell or nutrients available, again, it would not make sense for the cell to divide, right? Because then those cells wouldn't survive. So these are all things that the cell needs to consider before undergoing cell division. Okay, so moving on to the next section, which particularly talks about the chromosomes, right? The eukaryotic chromosomes. We're talking about eukaryotes throughout until, like I said, at the end of the chapter, I'll talk a little tiny bit about prokaryotic cell division. Okay, so it says here, the eukaryotic chromosome, right, contains a single DNA molecule, right? Double helix. We talked about the structure of DNA a little bit. And we'll talk about it again when we get to chapter 12, but it's that double helix structure, right? When we talk about chromosomes or chromatin, again, I mentioned this term before, that is DNA and protein, okay? So DNA is associated with particular proteins called histones, okay? And again, we call DNA and histones together chromatin, okay? These histones package the DNA, and we're going to talk about that. There's sort of different levels of packaging that occurs, but you can pack this huge amount of DNA into this tiny little nucleus because of the packaging that occurs due to these histones, okay? So so that's this part, right? They also play a structural role. It says they have essential survival functions, and then there are 5 different types. They're also involved in regulating transcription and how much transcription can occur. Remember, transcription is basically the cell making mRNA from DNA, and then that mRNA is going to end up getting translated into proteins. Okay, so histones are involved in that as well. Again, lots of detail that can be sort of inserted in here a little bit more. But let's just understand for now, for our purposes, that histones are proteins that are associated with the DNA that help package the DNA. Okay? All right. So like I said, there's sort of different levels, and we're going to talk about that. So it says DNA wound around an 8-histone core is called a nucleosome. Okay, so that's a name for a certain level sort of packaging. And again, it's just the DNA wound around a core of these histone proteins. I'm going to show you a picture. Okay, so that's the nucleosome. It kind of looks like a bead, and then you'll see there's these linker DNA sequence that look like a string. Okay, so there's like a nucleosome, a little string, which is the linker DNA, and another nucleosome, like a string, right? Another nucleosome. So like beads on a string. And again, I'll show you a picture in a second. That string then folds into a compact zigzag structure. Then it loops, and then it's loosely coiled. Okay, and then we're going to talk about chromatin at that point. Okay, so it says euchromatin represents the active chromosome, chromatin that can be transcribed. Like I said, right? I mentioned transcription. Okay, so the reason why histones are involved in transcription is because only certain areas of the DNA of the chromatin that's not wound so tightly would be able to be transcribed. Okay, so again, this is giving you an example there. I'll show you a picture. It'll make a little more sense in a second. Okay, so when we talk about chromatin, there are different types of chromatin. So euchromatin, active chromatin, it can be transcribed. It's not that tightly packed. However, there's more highly compacted form of a chromosome. It's called heterochromatin. So heterochromatin is inactive. These genes are hardly ever transcribed. Okay. And then we can talk about chromosomes, right? When you think about mitosis, right, and stages of mitosis, if you think about metaphase, and we're going to see a picture in a few slides, right, of these different stages, but those chromosomes, that look like kind of like X's, those are chromosomes, right? They're super compact, and you can actually see them as individual chromosomes. Okay, so it says most chromosomes have both euchromatin and heterochromatin. Okay. All right, so if you look here, right, here we go is what we talked about. We talked about beads on a string, right? So here's your nucleosome. Here's that linker DNA, right? And then you get formation of that zigzag structure, which kind of then forms this, we call this the 30 nanometer fiber. So this is all just different levels of packaging, okay? Then you have these radial loops that form, okay? So this is showing you kind of euchromatin here. And then this gets more tightly compacted, right? Sort of folds onto each other. And this will form the heterochromatin. And remember that then you have the sort of most compact or tightly packaged form of chromatin, which is the chromosome, right? So remember I mentioned the metaphase chromosome, right? And when we think about chromosomes, we think about them looking like this, okay? Very at this point they're very sort of tightly packed, and remember they're sort of composed of both euchromatin and heterochromatin. Okay, so now that leads us into talking about mitosis, right? So when we talk about mitosis, we're talking about nuclear division. We're following the chromosomes, right? And watching these and basically talking about the different phases in sort of separation of the chromosomes, right? Okay. And like I said, we will talk about chromosomes even more when we get to our genetics chapter. Okay. So kind of same thing I said at the beginning of this chapter, I'm going to talk about chromosomes again. And so I just talked about sort of chromatin and chromosomes a little bit here. We will again talk about it. Okay. So we're going to talk about it a little bit more here when we talk about mitosis, right? But we will talk about it again when we get to our genetics chapter as well. Okay. All right. So again, now we want to talk about mitosis, which is nuclear division, right? And then also cytokinesis, which is division of the cytoplasm. Okay. I already said this, right? Here, if we talk about cell division in general, both mitosis and cytokinesis. So during mitosis, right, those sister chromatids, right? Sister chromatids here, okay? Here's one chromatid, here's another. Okay, they are separated and distributed to those daughter cells. Okay, before mitosis begins, the chromatin condenses and coils into those visible chromosomes. And basically every species has a characteristic chromosome number. Okay, so here's a table showing you that. So we should be aware of how many chromosomes we have as humans, right? 46 chromosomes, 2 sets of 23. Okay, 23 from your mom, 23 from your dad. Okay, if you want to look at some differences in numbers of chromosomes, right? Yeast has 32 chromosomes. Okay, this garden pea, 14 chromosomes. Potato, 48. Okay, this type of fern actually has 1,320 chromosomes. Okay, so the number of chromosomes does not necessarily correlate with complexity, right? A goldfish has 94 chromosomes, human has 46. Okay, so it just has to do with how the DNA is organized, right? How those chromosomes are organized. Okay, so that was the other thing I'm going to point out, but it's on this slide too. Point out that this says diploid, right? So I said that humans have 2 sets of 23. So 46 is the diploid number. Okay, right? So that's what this is saying right here. 23 different types of chromosome. Okay, each type is represented twice. Okay, when we talk about haploid or N, so diploid 2N equals 46, 2 sets of 23. The only cells in the human body that have a haploid number of chromosomes are sperm and egg cells, right? So the gametes are the sex cells. Okay, because when the sperm and the egg come together to form that zygote, there's your 46. Okay, and so here again, I pretty much said all this, right? Makes a total of 2N equals 46 in each nucleus. One set from the individual's father, which we call paternal, and the other set from the individual's mother, which we call maternal. Okay? All right, so let's start talking about mitosis and the different phases of mitosis. So we talked about interphase a little bit, right? It says this, and we said the cell's preparing for division, okay? Organelles are duplicated, including something called the centrosome, because this is going to be important for mitosis, okay? It says here at the end of S phase, each chromosome is internally duplicated, right? Remember we said during S phase of interphase, S for synthesis or DNA replication. That's where DNA replication occurs. The cell is going to double its DNA, okay? So at the end of S phase, you have those 2 sister chromatids. Each chromosome is composed of 2 sister chromatids. Okay, they're genetically identical to each other. Okay, and they're attached at a single point called a centromere. I'll show you a picture again, okay, to show you the centromere and the 2 sister chromatids. During mitosis, the centromeres hold those sister chromatids together, but eventually they're going to separate, right? Then those sister chromatids are going to separate, and then each will become a daughter chromosome. And basically, you have these chromosomes distributed to each daughter nuclei, right? To each cell, okay? So that you make sure that each cell gets every single chromosome, right? It's not going to be good if, the cell has one cell has 2 of, chromosome 1 and 2 of chromosome 2, but then the other daughter cell is missing chromosome 1 and 2. That doesn't help. It has to have it has to have a copy of every single chromosome. Okay, so super important. You'll see that these chromosomes are lined up exactly the way they should be. Okay. All right, so this is again showing you this duplicated chromosome. Okay, so I always like this sort of fluffy looking chromosome here. So this is an electron micrograph showing you the chromosome. Okay, again, one chromatid, the other. Okay, one chromatid, the other. We call these sister chromatids. They are exact genetic copies of one another. This happened during S phase when the DNA was replicated. Okay, if this was not replicated, it would just have one arm. Okay, one chromatid. All right, so what this is showing you here, this is the centromere, right? That's where the 2 sister chromatids are joined together. Here is showing you the kinetochore, okay? And basically you have proteins that will bind to the kinetochore, and then that's where the microtubules will attach from the spindle, okay? So we're going to talk about that in a second. So again, These are sister chromatids. This is one chromatid. This is the other. These are exact genetic copies of one another. So every chromosome looks like this. It's duplicated, okay, before mitosis is going to occur. All right. So we mentioned the centrosome, right? So just outside the nucleus, there's a centrosome. This is the microtubule organizing center in animal cells. And I think we kind of mentioned this when we talked about the different parts of the cell, okay? So that centrosome gets replicated during interphase, right? So there's going to be two centrosomes because those two centrosomes end up forming the spindle on opposite ends of the cell, okay? So it says here, in animals, the centrosome contains centrioles, okay? And again, the centrioles, so the centrioles are part of the centrosome. In plants, there is no centrioles. That's why we're pointing that out, saying in animals. But basically, the mitotic spindle is going to form from those, okay? So that's what this is telling you here. Right, you got the microtubules. They assemble, right, made up of tubulin, and basically they're gonna allow for the change in cell shape, but they're also going to be able to grab onto those chromosomes and pull them to opposite ends of the cell. So we're going to see pictures of this. It will make sense. Okay, so let's talk about the different phases of mitosis. Now, I will tell you that the book breaks down the phases into a few sort of kind of intermediate phases, right? So it'll break it down. Yeah, so even here, it's not even showing you. So in the picture, let me just tell you what I mean here, right? In the pictures, the way it has it broken down, it talks about early prophase, right? And then later prophase, then prometaphase, then metaphase, anaphase, and telophase. A lot of times we just talk about prophase, metaphase, anaphase, and telophase. So on the lab quiz, like if I show you guys a picture like this, your choices will probably just be, they will be prophase, metaphase, anaphase, or telophase. Okay, so I'm not necessarily going to ask you to distinguish between early prophase or prophase from the pictures. Okay, but the reason why it's sort of divided up this way is just because it just allows you to be a little more specific in terms of what's happening earlier on in a phase versus later. Okay, so I just wanted to point that out. All right, so if we talk about prophase first, right? Again, this is the very first phase of mitosis. So nuclear division is about to occur. Chromatin are condensed, right? So that now you can actually see those individual chromosomes, okay? Because they're highly, highly condensed. Like we talked about that. Each chromosome has two sister chromatids. They're held together at the centromere. Again, we talked about that as well. What else is happening? The nucleolus will disappear. The nuclear envelope will start to fragment. The spindle will begin to assemble, okay? The two centrosomes will move away from each other because they're going to start moving to opposite ends of the cell. And in animal cells, you can see these asters that form, okay? All right, so again, I'll point all this out in the pictures again. So let's just kind of go over it in words. It's kind of the same deal. Those pictures, if you look at the pictures and the description that's there, it has all the information you need. The slides are sort of just emphasizing that, okay? So now if we move on to prometaphase, so again, this is sort of this intermediate phase between prophase and metaphase, okay? It says preparations for sister chromatid separation are evident, right? The centromere of each chromosome develops two kinetochores. I told you the kinetochores are where the microtubules are going to grab onto. And so the kinetochores are made up of proteins, okay? And again, there's one attached to each sister chromatid. So that was this, right? Here's the kinetochore here, okay? So it says they physically connect the sister chromatids with the microtubules, right? Okay. If we move on to metaphase, here metaphase middle, metaphase plate, this is where the chromosomes line up in the middle of the cell, okay? So chromosomes are pulled around by the kinetochore fibers, and they basically line up in the middle of the cell, okay? And again, we call that the metaphase plate. It says non-attached polar spindle fibers overlap, and that helps sort of elongate the cell and change cell shape. I'll show you what I mean by that in a second. It says M checkpoint delays start of anaphase until kinetochores are attached properly. So remember that checkpoint I pointed out, that spindle assembly checkpoint is going to check to make sure the spindle is properly formed, right? And that all of the microtubules have grabbed onto those chromosomes, okay? If not, then the cell will not proceed through mitosis. All right, next phase is anaphase. Okay, the centromere will dissolve. Okay, the sister chromatids will be released. The sister chromatids they separate at the centromere, and now basically you're pulling each sister chromatid to opposite sides of the cell. Okay, so as I was saying, in anaphase, right, the centromere will dissolve so that those sister chromatids can separate from one another. Okay, and then they start to get pulled to the opposite ends of the cell. Okay, and now they're called daughter chromosomes because they're now separate. All right, and it says the poles move further apart. I'll show you all this in the picture as well. So in anaphase, the chromosomes have this sort of V shape, right? The chromatids as they pull away from each other. All right, the last phase of mitosis is called telophase. Okay, the spindle disappears because now those daughter chromatids or chromosomes have already moved to the opposite sides of the cell. There's a new nuclear envelope that's forming around those daughter chromosomes, okay? And so you have these two clusters of daughter chromosomes, okay? And basically, like I said, the nuclear envelopes form around, and basically each one of those is going to become a separate cell, okay? So you need to have this cytoplasm and all the organelles divide. That's the cytokinesis part of it. Okay, then those chromosomes start to become less condensed, right? And they look like chromosomes again. They don't look like these individual chromosomes, okay? All right, yeah. So like I said, all of these pictures also describe what we just went over as well. So probably when you're studying, it's better to study from the picture so you can see what's happening and also kind of see it in words, all right? And again, on the lab quiz, I will show you pictures of either a plant cell or an animal cell and say, what phase is this? Is this prophase? Is this metaphase? Is this anaphase? Or is this telophase? Okay. We also should be able to identify the difference between an animal cell and a plant cell just by looking at that picture. Okay. All right, so I'm probably not going to read through every single thing here because I went through that on the previous slides, but I'll sort of hit some high points here. But again, this is early prophase. Centrosomes is duplicated, okay? Chromatin is condensing. Nuclear envelope is fragmenting, okay? And you can see that here as well, okay? You can see these asters. Remember we mentioned asters forming because it's basically these microtubules starting to come out here. You don't see that in plant cells, okay? All right, we move on to later prophase, right? So now it's showing you the nucleolus has disappeared, right? Pretty much, you know, that nuclear envelope is pretty well fragmented, okay? Now you can see these separate duplicated chromosomes, right? As individual chromosomes, you can see the two sister chromatids. You can see how they look a lot different than they did here, right? This is sort of more diffuse looking, kind of like a bowl of spaghetti, where these are more condensed and sort of more distinct, okay? So you can see that also in the picture here. You can also see that these centrosomes, right, have started to move apart, okay? Right, and so here again, you can see how these chromosomes look different. All right, now, and so notice that in the plant cell, you don't see the spindle because you don't have those centrioles in the aster, so it's very hard to see. All right. Now, if we get to prometaphase here, so this is just before metaphase, the end of prophase essentially. Again, you can see that you have these distinct chromosomes with two sister chromatids. You have the kinetochore that's formed. The this spindle is pretty much, you know, you can start to see it forming. There's poles, right? They've moved, so it says spindle pole because basically these centrosome, right, have basically the two centrosomes have moved completely apart from one another and they're on opposite sides of the cell. And again, more and more of these microtubules are going to start to come out of that, okay? All right, if we move on to metaphase, okay, Centromeres of duplicated chromosomes are aligned at the metaphase plate, right? We can see them all aligned at the metaphase plate. So basically, if you see a picture like this where you can see these dark chromosomes all lined up in the middle, we know it's metaphase. It's one of the easier phases to identify. And again, you can see the spindle here, right? And if you look at this cartoon, you basically have some of these microtubules, right? These kinetochore microtubules that basically grab onto the kinetochore part of the chromosome, right? We talked about. Then you have other ones that don't grab on. Those are just going to help elongate the cell. Okay, so this is what this says here. Kinetochore spindle fibers attached to the sister chromatids come from opposite spindle poles. Okay, so like this one's going to attach here. This one's going to attach on this side so that eventually they're going to be able to pull this sister chromatid this way and this one this way. So based on the way they line up is the way the sister chromatids are going to go. All right. So, all right. So again, make sure we can identify from a animal cell and also from a plant cell. All right. So the other thing to note, just when you're looking at these pictures, blue is representing paternal, red is representing maternal. The size here, so there's 4 total chromosomes in this example cell. Two sets of 2. Okay, just to kind of point that out so we can understand those things and how to follow them. So basically, this little blue one and this little red one, right? We can say that those are chromosome 1, right? 2 copies of chromosome 1. These bigger ones, this is chromosome 2. Okay, 2 copies of chromosome 2. So now you can see when they line up, you want to make sure you get one of the paternal chromosome 1, one of the maternal chromosome 1, right? One of the paternal chromosome 2, and one of the maternal chromosome 2. Okay, so you can see how you're getting every single chromosome to basically move to the opposite ends and then become part of the daughter cells. Okay, so now we're in metaphase. Sorry, I wanted to make sure I pointed that out. Okay, so we're at metaphase. The chromosomes are all lined up on that metaphase plate. What's going to happen next? Those kinetochore, remember we said those spindle fibers, the kinetochore spindle fibers grabbed onto the kinetochore there, and now they're going to pull those sister chromatids apart. At this time, we call these daughter chromosomes. Okay, so again, if you look at the microscope picture here, you can see how these are being pulled apart. Okay, daughter chromosomes. You can see the same thing in the plant cell. In the plant cell, sorry, I didn't kind of point this out here. When you look at metaphase, you can see again them lined up. Now you can kind of see the spindle right in the plant cell at this phase, but again, it's just a little harder to see at the earlier phases because you don't have the centrioles and the aster. Okay, but now you can kind of see those spindle fibers. All right, anyway, anaphase, right? These become now daughter chromosomes. They're separated. Okay, they move towards each pole, and you can sort of see too, like the center of the cell is starting to kind of start to pinch together. Final phase, telophase, right? Now these daughter chromosomes have moved to opposite ends. You see the spindle has gone away, right? The nucleoli start to reappear inside here. The nuclear envelope forms around each set of daughter chromosomes. Okay, it's showing you here this cleavage furrow that's starting to form. And basically, the last thing that happens is cytokinesis. So the formation of this cleavage furrow is really part of cytokinesis. And what's going to happen, I'm going to talk about it in the next slide, is it's going to pinch this into 2 cells, right? So now you have 2 daughter cells, right? These are exact copies of that original cell or parent cell that you started with. Okay, so like I said, cytokinesis is the division of the cytoplasm. It will allocate the mother cell cytoplasm equally to the daughter cells, also including, making sure the organelles that need to be in each daughter cell are there as well. It says it often begins in anaphase, right? So it's kind of something that's happening sort of towards the end stages of mitosis, but remember, it's not part of mitosis. Okay, animal cytokinesis, I pointed that out. Of cleavage furrow forms, okay? And again, that kind of squeezes it, right? So it says it's formed by a contractile ring of actin filaments, and that, again, pinches it into 2, okay? So let's see what it says, like pulling on a drawstring. In plant cells, however, though, there is no cleavage furrow. Imagine a plant cell has a really rigid cell wall. So there's no way that the cell would be able to separate into two by squeezing it. Okay, so what forms there is something called a cell plate. And let me just show you. You can see here it's showing you the formation of the cell plate there. All right. So this is basically, there's actually these sort of membrane-bound vesicles that make up that cell plate. Okay, and they basically end up fusing together. Okay, and so it becomes this sort of plasma membrane between the daughter cells, and then basically that sort of then ends up being a cell wall. Okay, so you don't really need to know these specific details here about this middle lamella. You don't need to know about the middle lamella and all that. Basically, just know that it's a cell plate that forms between the two plant cells, because then eventually the cell wall that will form. No way a cleavage furrow would work in a plant cell situation because of that rigid cell wall. Okay, so if you look more at this contractile wing, and this is actually a real electron micrograph showing you that process of cytokinesis, showing the cleavage furrow that's formed, and then basically a sort of squeezing that occurs due to this contractile ring and the separation into two. Again, if you look at plant cell cytokinesis, basically this plasma membrane, this cell plate will form first, and then eventually it will become a cell wall. Okay. All right. Now, we've been talking about, the stages of mitosis. We talked about cell cycle. Let's talk a little bit about why the mitosis is important, right? And functions of mitosis. Okay. So in terms of, general functions, growth, repair, right? If you're going to grow or you're going to repair tissue, you're going to need to make more cells, right? Think about that. If we talk about plants, plants have the ability to continue to grow throughout their entire life, okay? So they have this special tissue called this meristem or meristematic tissue that basically retains that ability. Those cells keep dividing, okay? So you always have this growth that occurs. Now that's not true, obviously, in animals, right? They reach a certain size and then they stop, but mitosis is still going to occur for repair, right? And things like that. Okay, so super important no matter what. In terms of like skin cells, right? Your skin cells are replaced all the time, and so there's always mitosis going on in that tissue. If we think about development, right? It says in mammals, mitosis is necessary when a fertilized egg becomes an embryo, right? Fertilized egg becomes an embryo, right? Well, I mean, we even think about a fertilized egg becoming a zygote, then an embryo, right? Embryo then becomes a fetus, and then child becomes an adult, right? These are all things that I just talked about. So, but when you talk about just development in general, think about going from one single cell, right, to a fetus. That's a lot of that's due to cell division. You have to have tons and tons of cell division that occurs, and then you also have to have development and differentiation into specific tissues or what have you. But mitosis is super important in terms of having enough cells there to form that embryo and to form that fetus. Okay. Also, again, if we're just talking about growth from a child to an adult, mitosis. Okay. And like I said, repair, right? Super important too. Healing of a cut involves mitosis. Okay. Healing of a broken bone, again, involves mitosis. Okay. All right. So another thing to kind of point out that's sort of part of this, and you guys don't need to know details about therapeutic cloning and reproductive cloning, but I guess it's just something interesting to mention here. So it says here that many mammalian organs contain stem cells. So remember that meristem tissue that I mentioned in plants? Well, it's the same kind of idea. There are, certain cells that retain that ability to divide, okay, in animals. And those are the only cells that are able to keep producing more cells, okay? Other cells that don't have these stem cells, they won't be able to make new cells. So like neurons, okay? They don't really actively divide, right? There are actually, I shouldn't even say this, but anyway, I'm not trying to confuse you, but there are actually two regions of the brain where there are some stem cells that are there. And in those regions of the brain, the body can make new neurons, okay, because of that stem cell population. I guess the point is that you have to have this stem cell population to retain that ability to divide, okay? Gives you an example here of red bone marrow stem cells, okay? They continually produce blood cells throughout life. And if they stop doing that, leads to a major problem. Okay, so those stem cells are maintained throughout life. All right. Now, we can exploit these processes, right, to do things like cloning. Okay, so again, I'm not going to hold you guys responsible for the details. This is something that we might do as an extra kind of thing in class. So just worth mentioning. I'm not going to ask you about it on the exam, right? But if you talk about therapeutic cloning versus reproductive cloning, right? So therapeutic cloning is basically cloning to produce human tissues, okay? And you can use stem cells, like I mentioned, these bone marrow stem cells, right? Or you can use embryonic stem cells. And then you can basically put them in an environment, in a dish, and induce them to become a certain type of tissue, right? Like muscle tissue or nervous tissue, okay? And so that just produces the tissue, right? Or more cells in that differentiated state. If you talk about reproductive cloning, this is actually making a new individual, right? So like the sheep dolly that was cloned. So we do technically have the ability to clone whole organisms. I don't think we have it exactly right right now, but we have that ability to do that. And the other question is sort of, there's a lot of ethical concern here in terms of, well, then what do we do with that ability, right? And is it something that we should even think about doing? Okay. So this picture just shows you this reproductive cloning here, making a new individual, and therapeutic cloning. Again, you don't need to know these details, okay? Just something that's interesting and worth noting. Okay, another thing that's super interesting and worth talking about, and again, we're not going through, all of the details here, but understanding the relationship between the cell cycle and cancer. And I sort of alluded to it when we were talking about the cell cycle. We all have heard of tumors, right? We all know that cancer is the sort of overgrowth of cells, right? So if we're going to think about just abnormal growth of cells, in a particular area, we call it a tumor, right? You can have benign tumors that are not cancerous, right? So these stay encapsulated. They don't invade neighboring tissue or spread, okay? But however, if we're going to talk about a malignant tumor, those are cancerous, right? Not encapsulated. Those cells will readily move into a neighboring tissue and then can also get into either lymphatic vessels or blood vessels, and then they can go to very distant places in the body. That's when you're in trouble, right? And we call that metastasis. Metastasis, okay? And so this says it results from mutation of genes regulating the cell cycle. Okay. Again, imagine you have a cell that should not be making a whole bunch of new cells, but for whatever reason, because of a mutation, it does, it leads to the formation of tumors. Okay. So the regulation of the cell cycle is super, super important in the development of cancer. Okay. And in fact, many genes that are implicated in cancers, like things like BRCA, right, we've heard of maybe, or other types of genes that, again, have been shown to be related to specific cancers, they are involved in regulating the cell cycle. Okay, so if you're going to study cancer as a scientist, you need to know a whole bunch about the cell cycle and all the things that regulate the cell cycle. All right. If we want to just talk a little bit about, in general, sort of development of cancer, right, in terms of some of the steps that occur, one thing to note is that it's not an instantaneous thing. It's gradual. It's multi-step. I sort of mentioned that there are, genes that have been implicated in certain cancers. Well, there are many, many genes that are mutated in particular cancer. You can't target it to one gene, right? It is not a purely genetic disease or anything like that. So that's why it's gradual, why it's multi-step. So it could be years before a cell is obviously cancerous, but it probably had problems, sort of all along. But once there's enough problems, it leads to that cell becoming cancerous, okay? And so if we talk about some characteristics of cancer cells that you see, here are some of them. Okay, so cancer cells have the tendency to form tumors. They form tumors, right? It says mitosis is normally controlled by contact with neighboring cells. So something called contact inhibition. Cancer cells have also lost that contact inhibition, and I'll show you a picture in a second. Meaning, usually, if a cell in a given tissue, if they start dividing, they'll know that it's time to stop dividing because there's too many other cells around. Cancer cells don't even have that, right? They just keep dividing, even though there are lots of other cells around, okay? They also lack differentiation, okay? Meaning they're non-functional in their tissues. So like if you're talking about like, you know, a lung cell or something like that, these cancer cells are just there and keep dividing. They don't actually function as lung cells, okay? They have abnormal and large nuclei if you look at them morphologically. We mentioned this before, they undergo metastasis, and angiogenesis is actually the formation of new blood vessels, okay? So they can actually form their own new blood vessels, and that helps them get nutrients and oxygen so they can continue to divide. So these cancer cells are pretty smart, right? They sort of they can override these regulatory processes, and then they can also do things to actually make it better for them to divide. And then also they don't undergo apoptosis, right? Which is cell death. Remember we said that sort of mitosis and apoptosis are sort of opposing forces, all right? So again, even though there is something wrong, the cell will not go through programmed cell death. All right, and so this is just showing you some kind of the progression of cancer here, and points out some of the things I just mentioned on that last slide. Okay, so this is showing you, you know, a certain tissue here. Here's the one cell, this brown cell that has some sort of mutation, right? And it has the ability to start a tumor. Okay, and so this is this primary tumor that's formed by this brown cell because it's going to basically divide out of control. And then what you now see here, there's this another purple cell within this tumor that ends up mutating even more, right? Because again, these are rapidly dividing, they're overriding all these regulatory processes, so they form mutations more rapidly than normal cells. Okay, and now what you see is this purple cell now forms, you know, more of the tumor, and more of them become purple cell-like, and now what do they have the ability to do? Metastasize, right? And move on to distant places and form tumors in other places as well. Okay, so you can see that it sort of starts out from a mutation, right? And then it progresses further, okay? All right, so this is what we mentioned, these characteristics of cancer cells versus normal cells. Okay, so be familiar with these, understand this, right? These are things that we talked about already. Okay, all right. Couple more slides on this, and again, this is a lot, but I think it's again worth going through. In terms of, again, origin of cancer, we said it's sort of multi-step, right? And so this sort of illustrates this. So it says normal growth and tissue maintenance depends on the balance between signals that promote and inhibit cell division, right? So again, this is the idea that if the cell cycle is not properly controlled, it can lead to cancer, okay? And we said that it's due to mutations, right? I just showed you this picture where we see this mutation and it forms a tumor, right? I said there's many genes that are implicated in certain cancers, okay? So what are those types of genes, right, that are mutated? What do we call them? So there's two sort of names here. So oncogenes, right? These are sort of, again, two types of genes, and most of oncogenes and tumor suppressor genes, they're involved in regulating the cell cycle. Okay, so these are all cell cycle genes. That's kind of what we said. But if we want to be a little more specific, right? Oncogenes, okay, are the cancerous forms of the genes, but proto-oncogenes are normal genes. Okay, these are normal genes that exist. They code for proteins that promote the cell cycle. There are many times that the cell cycle should be promoted, right? We talked about growth and repair and things like that, okay? However, if one of these normal genes that help regulate the cell cycle in this way become mutated, right? And that gene's always on and always promoting the cell cycle when it shouldn't, it becomes an oncogene. Oncogenes can cause cancer, okay? And again, it's not just one specific gene, right? Like if I have a mutation in this one specific gene, I know I'm going to have breast cancer. That's not how it works, right? It's usually a mutation in one of these genes and maybe other genes, and then some sort of environmental factor that comes along. It's all of these things play a role together, okay? And that's why it's so hard to treat cancer or to come up with a sort of end-all be-all treatment, because all these cancers are so different and there's so many different things that happen, right? It's a multi-hit type of disease, which is true for many other diseases, but particularly for cancer. On the flip side, if we think about tumor suppressor genes, these are normal genes, right, in your body, in your cells, that code for proteins that inhibit the cell cycle or slow down the cell cycle or may induce apoptosis, right? Because again, this is a normal process. This should be happening, right, if necessary. If one of these genes becomes mutated and then those tumor suppressor genes aren't functioning at all, it then can lead to cancer, right? Because if a tumor suppressor gene is normally supposed to slow down the cell cycle, but a mutation has occurred to make it ineffective, it will not be able to slow down that cell cycle, right? And therefore can lead to cancer, right? So you can see these are kind of two different types of genes and also kind of mutations that have different effects on them can lead to cancer, okay? So for this next thing, you don't need to know specific examples here. So this is just giving you an example of some genes, right? Tumor suppressor genes, okay? So if you're interested in knowing some examples, and there are many others as well, two examples here, retinoblastoma, RB, and P53. These basically, so retinoblastoma is basically an inherited cancer, a child cancer of the retina. And basically it can be specifically pinpointed to this retinoblastoma gene, okay? And then P53 is also involved in this sort of pathway and turns on other inhibitory genes, okay? It says half of human cancers involve an abnormal or deleted P53 gene, okay? So this is the gene that you see mutations in a lot of different cancers. Not only that gene, other ones as well, but for instance, if you went for genetic testing, I guess, and you had a mutation in the P53 gene, clinician might tell you are at increased risk for the development of cancer. Does that mean you're going to develop cancer? No, but it may put you at an increased risk, okay? All right. I just want to see something here. I want to see if I have a picture of that. Yeah, I really don't. Okay, so let's just take this as these are some examples of tumor suppressor genes, okay? Again, tumor suppressor genes normally will slow down the cell cycle. You can have a mutation in a tumor suppressor gene that turns it off and therefore causes the cell cycle to be increased, right? Causes more cell division to occur, okay? All right. Okay, sorry. So this is just throwing me off here. So maybe this will throw you off too. It says here, examples are the retinoblastoma, RB gene, and P53 gene, which code for proteins with the same names, meaning retinoblastoma codes for the retinoblastoma protein. This codes for the P53 protein, not that they code for the same protein. Sorry, that's what was throwing me off because I was like, that doesn't make sense. These, again, are just examples of tumor suppressor genes. Remember, we also mentioned proto-oncogenes, right? And that proto-oncogenes can be mutated to make them more active. And then that causes cancer, right? An example of a oncogene is this BRCA gene. I mentioned this. So there's a specific, and you can get tested for this, genetically tested for this particular gene, again, tied to certain breast cancers, okay? Again, if you test positive for the mutation in this BRCA gene, does it mean you're going to get breast cancer? No, but it puts you at an increased risk, okay? So again, these two slides just give you a little more detail on oncogenes and tumor suppressor genes and give you some examples, okay? All right, so like I said, just a little more info on proto-oncogenes and what they do, okay? I sort of told you on that first slide that they are genes that stimulate the cell cycle. They promote progression through the cell cycle. A little more detail here. All right. So it says here there are 100 oncogenes which can lead to tumors. I'm pretty sure we probably know of more than 100 at this point, okay? So we continue to identify more and more oncogenes and tumor suppressor genes, okay, as we, sort of understand the genetic basis of cancer a little bit more. All right, now this slide is basically kind of showing what I was basically kind of pointing out this whole time, or sort of explaining that you have multiple influences that can then lead to cancer, right? So this is saying heredity. So meaning having mutations in a tumor suppressor or an oncogene, right? Or a proto-oncogene like we mentioned. Exposure to radiation can then cause genetic defects that can lead to cancer. Exposure to pesticides, herbicides, right? A virus. There are many cancers that are triggered by viruses, okay? So these are the sort of environmental type of hits that can happen, okay? So again, it's not any one combination of things. It's different in different people, but understand that it is a combination of things. It is a combination of genetics and also environmental exposure. You can add in stress here, right? Any of that can contribute to that as well, okay? So, and then this diagram is just showing you a little bit in terms of kind of example of kind of what a proto-oncogene might code for versus what a tumor suppressor gene would code for, right? Types of protein here. So it gives you just a little bit more detail again about that. Again, I just want you guys to understand what a proto-oncogene is, what a tumor suppressor gene is, and what would happen or what types of mutations in these genes could lead to cancer and why. Okay, definitely probably an essay question on the next exam about that, right? And meaning me sort of asking you about that, asking you to explain that in your own words so I could see if you really understand that. It's not about knowing all these specific details or memorizing any of them or anything like that. Okay. So anyway, so like I said, this is really meant to kind of put together everything that I was saying in the last couple slides. It's not meant for you to kind of, memorize what's going on here. Again, I was explaining this as we went along. And so we want to understand sort of the development of cancer, right, in general, and understand that it's a combination of genetic factors, i.e. mutations in proto-oncogenes, mutations in tumor suppressor genes, and then it's also environmental influences, okay, that can affect the cell. Things like radiation, things like pesticides, things like a virus. It just depends, okay? All right. He mentions also here, says other causes of cancer, telomeres, okay? So there is something that basically every chromosome has at the end of their, kind of at the end of the DNA, end of the chromosomes are called telomeres. And each time the cell divides, these telomeres get shorter just because, and we'll talk about this when we talk about DNA replication, but just because when the cell replicates its DNA, it cannot replicate the extreme ends. Okay, so these telomeres don't code for proteins or anything like that. They're not genes that code for anything, but they keep getting shorter and shorter. And if they get too short, okay, or as they get shorter, it signals the cell that they shouldn't divide anymore, right? Because if they divide again, they may very well be missing DNA that they need, okay? There is an enzyme that will maintain the length of these telomeres, okay? So there's that, right? So the cell wants to maintain the telomeres at a certain length. However, if they start to get too short, it signals to the cell that, oh, maybe I shouldn't divide again because there's a chance that I may not be able to replicate all of my DNA, okay? There are mutations that can occur in the telomerase gene, okay? And so it says it causes telomeres to continue to lengthen, which allows cancer cells to continually divide. So basically, again, a mutation here can keep those cancer cells from dividing, right? When otherwise normal cells, they would be triggered to stop dividing. But because the cancer cells have a mutation in this telomerase gene and sort of have overridden that one regulatory kind of point where if the telomeres get short, it shouldn't divide, the cancer cells will be able to keep dividing, okay? And lastly, the last thing to mention is I told you the very last section was prokaryotic cell division, and really I don't really talk about it much. We just want to know that prokaryotes like bacteria do not divide via mitosis. Okay, they don't have a cell cycle. Their division is called binary fission, and really it's just splitting into two. Okay, so, and also understand that their DNA is much different. It's just a ring of DNA. Okay, it's folded up in an area called the nucleoid. It's about 1,000 times the length of the cell. Basically, it gets replicated into two rings, just like we talked about replication in eukaryotic cells. But those two rings, it gets replicated into two rings, and then basically those two replicated rings attach to the plasma membrane, and that's how the cell keeps it apart. And then you basically have the splitting into two. Okay. The one thing though about binary fission and cell division in prokaryotes is that this makes two new individuals, right? So it's a form of asexual reproduction. When we're talking about eukaryotes, or we're talking about, in humans, particularly in animals, it's not a means of reproduction, right? It's just a means of producing more cells, cellular reproduction, but not of a whole organism. Okay. In terms of, asexual reproduction, if we talk about E. coli, right, a type of bacteria, generation time 20 minutes, meaning every 20 minutes you can produce more and more bacterial cells, okay, and new organisms, right, single-celled organisms. Okay, so if you want to sort of compare, right, binary fission in prokaryotes to mitosis in eukaryotes, these are some things to point out. Okay, binary fission and mitosis both ensure that the daughter cell is genetically identical, right? It says prokaryotes, right, are single-celled. And so basically cell division of single-celled organism produces two new individuals. There are also other types of eukaryotic cells that are single-celled, like yeast, and that then can be a new individual, and again, asexual reproduction. So it's not only in bacteria that you see this, you can see this in other single-celled organisms as well, okay? So that's the idea here. Right? Multicellular fungi, plants, and animals, it's growth, renewal, and repair. We already said that. Okay. But again, remember that this cell division can be a means of reproduction if you're talking about a single-celled organism. An example of a single-celled prokaryote bacteria, but there are also single-celled eukaryotes as well. Okay. All right. It says here, binary fission, chromosome duplicates, each daughter cell receives one copy as the parental cell elongates and a new cell wall and membrane form, okay? No spindle involved in binary fission. All right, so if you take a look here, you can see this happening again, much more simple than mitosis, right? So you basically have that attachment of the chromosome to a special plasma membrane site indicates that it's ready to divide, right? Then it replicates that chromosome, right? And then basically you have the cell wall and the plasma membrane that's going to form between the two, and you get two new daughter cells, okay? You don't need to know the exact steps here, and in fact, there's not that much detail here anyway. So I'm not really going to ask you about the different phases of binary fission. I'm not going to show you a picture of a prokaryotic cell and say what's happening here or anything like that. So you don't need to identify that like you would for the phases of mitosis. Okay. In terms of functions, we already said this, but here it is in table form, right? If we're talking about bacteria and archaea, binary fission, it's a form of asexual reproduction. In eukaryotes, can be a form of asexual reproduction, but if we're talking about multicellular organisms, it's development, growth, and repair. All right, and that concludes that. So rather long lecture, but super important. Lots of important concepts in this lecture. Remember, for the lecture exam, I'm not going to show you pictures of the different stages of mitosis to identify, but for the lab quiz, I will. There will be both pictures of plant and animal cells that look very similar to the pictures that were in this PowerPoint, okay? And will look similar to some of the pictures that you see when you do your labs, okay? So being able to identify. For lecture, though, you need to know what's happening at each one of these phases, right? There will be questions that ask you, at what phase do the chromosomes line up along, the middle of the cell or the equator of the cell, okay? So those types of things. All right, guys, thank you.